Alright, in this video we're going to talk about what is a gene. A gene has been defined in many ways over the last, say, century and a half, when people started realizing that there were traits, characters that could be inherited from one group to the next, from one generation to the next. So they came up with a term called a gene, a unit of heredity. And so we're going to start with that concept of a unit of heredity. And I would say in the last, I'd say from 1950s to the 1990s, a pretty good definition was this open reading frame, or this promoter to terminator, which we've already learned about. That was the general definition. But now we know some more things once we sequence the human genome, and that definition is changing. So let's look at this. This is our general picture. Let's see if I can get this a little larger. Nope, let's fit. We can get that maybe to 50. There we go. This is a representation of a gene that we've already learned about. Here we unwind the DNA, and we're going to make a copy of it. Remember Grandma's cookbook, our copying machine. And our copy is the mRNA, which would be right here in red. Now here, all right, is our promoter. Here is our terminator that we have talked about. And the idea is that the RNA polymerase, a protein, is attracted to this promoter. And we learned about a particular promoter, which was TA. Oops. Just really hard to write on this thing with a mouse. But TATA will be right in here in this light blue area. So the letters TATA are, when they're next to each other, are going to attract this particular protein to attach and stick to this particular piece of DNA. That's our copy machine. And what's going to happen is that it's going to make a copy. It's going to make this RNA right here, RNA transcript that we see here. And it's going to continue to make that copy until it hits a terminator. And the terminator, if you didn't realize it, was our hairpin loop that we learned about in a different video. And that hairpin loop, if you remember, had like A's or T's on one side. And then it had G's and C's up here in the loop. And the thing is, it's single-stranded. And it's actually the RNA that ends up forming the loop. But the letters are going to be, since it's complementary, the letters, you can find it by just looking at the letters, because you know that you get a bunch of A's and T's together, and then you get, and in between them are some G's and C's or whatever. Anything that will actually form this structure is going to be a terminator. So the idea is that this piece kind of bonds with this piece, and go make sure you go watch that video before you move on if you don't remember about the terminator. But the whole idea is this causes our polymerase here to fall off. So it attaches here and moves across, and then when it hits this region, it falls off the DNA. And when it does, you have completed your RNA transcript. And so for many years, the general idea was that this region here, the coding region, was considered the gene. All right, and we have recently added, not recently, but in the, say the 90s or so, 80s, 90s, they started adding the promoter and the terminator to our definition. And so you get something like this. They call it a transcriptional unit, and that's a gene. All right, and this is just a representation of the same thing we just saw. You do need to understand that on this side you have what's called upstream, and upstream is before the point where the copying starts. So the RNA polymerase will attach here at the TATA, move in this direction, all right, and fall off here when it gets to the terminator. And the region in here where you start copying is called the transcription start site, and anything before that is upstream, and anything after that is downstream. And you need to know those terms because I might use them on a test question. So the idea is just, this is what for the most part has been considered a gene, and in many cases this is still what's considered a gene in most textbooks, but we're finding that there's way more to it than that, and probably this definition is going to have to change. Now, what ways is this going to have to change? Well, we're going to look at these other parts we know what the open reading frame is. That is going to be what we call the part that gets copied, all right, into a protein. I should say it gets copied into an mRNA, which then is read by the ribosome, if you remember Grandma's cookbook, and then those are the instructions for making a protein. And the ribosome will, of course, put together a protein with those instructions. We then learned about the promoter and terminator. We now need to learn about this, the CpG island. So let's move back here to our, let's move to the next slide. Let's see, here we go. All right, CpG islands occur out here in the promoter region. So think of your TATA box as being in here somewhere before you start your gene, before you start your open reading frame. All right, you have, okay, if this is the open reading frame as we were calling it, we have the TATA right behind it. The terminator would be over here. And what you have are this something called a CpG island. A CpG, C followed by a G, CpG, is just saying that we have some DNA, and that DNA on one of the strands, actually it's going to be both because they're complementary, you have a C here followed by a G. That is a CpG, and that is different from a C with a G being over here. C over to here is a hydrogen bond, so actually you could go CHG because this is a hydrogen bond. This is a phosphate in this bond here. It's a little stronger. It's a covalent bond. So it is a CpG going this direction. So that's why we call this a CpG island. C's followed by G's are very rare in the genome. And so anywhere they appear, especially in clumps, we call it a CpG island, and they tend to occur right before genes. And so all these C's followed by G's in the promoter region, all right, allow you to do something. What it does is a protein, another type of protein that we won't even name it, can attach to C's and G's. Just like the RNA polymerase, it will attach here and it will what we call methylate it. It alters the C's in the C's and G's. And this methylation, here we have them in blue, that methylation will actually turn the gene off. We will talk in another video about how it does that. But when this protein, that we'll rename them, no need to even name it, there are actually several of them, but they will essentially methylate a CpG island outside of a gene, and that will essentially turn it off. This is an on and off switch. If it's unmethylated like this, all right, then the gene is on, and RNA polymerase will go and copy it. If it gets methylated, RNA polymerase will not attach, will not move across the open reading frame, and it will not make the RNA, so you don't get a protein. So keep in mind what a CpG island is, and this is kind of like your on and off switch. This is the piece that you make the copy from, but we also have in our promoter region, out here where the TATA is, and actually a little bit into the gene, what we call the promoter region, you can have a CpG island, which is an on and off switch for your gene. All right, so that kind of covered this, but then when we actually sequenced the human genome, we realized we really didn't know much about what was going on. We learned, actually this is just showing the open reading frame. I'll explain that in a different video someday. Oops, I have previous writings on this. Let's move here and let's erase all of this. All right. We have this weird case that I'm about to show you here. Let's get most of this off. We don't have to get it all. Get it down here around the gene and get that off. Now, what we need to realize is that here is our promoter sequence. So our TATA would be somewhere, say, here. That's our TATA and our promoter somewhere in there, right here. And we'd have a CpG island in here somewhere. So a bunch of C's and G's. But we also have this thing called an enhancer sequence, and we have a protein that sticks to it, or maybe even multiple proteins. And what happens is this sequence, just like these, the TATA, which attracts proteins, so does this enhancer. This enhancer has a sequence that attracts certain proteins. And certain proteins will bind here, and what happens is the DNA will bend, and it will bend over here and touch your RNA polymerase. So here is our RNA polymerase, and you can see that the DNA has bent over, and you have a bunch of proteins, and there are some others here that are important, and they allow this to attach, essentially. When this happens, it can either keep transcription from occurring as fast, maybe slow it down so you don't get as many RNAs made, or it can potentially speed it up. So this one is actually does it say activator? I can't quite make it out. Let's kind of get a little, oh, that's the wrong direction. Activator protein. So here, this upstream enhancer bends over and speeds up transcription, but it could just as easily slow it down. So this particular type of system is a volume control. So in other words, it controls not on and off like the CpG island, it controls volume. It controls how many RNAs are made at any given time. So you now have something else that can affect transcription, but it controls the rate or the volume. So the rate of transcription is now controlled by whether or not this sequence is here and whether or not a protein is bound to it. So it might be here, but depending on which protein binds here, it might speed it up or it might slow it down. Now there is another thing that we need to know about. There's usually a bending protein that can attach here. Now we call these proteins that bind in this region, anywhere before transcription starts. Any of this region, the promoter region or enhancer region and the extended regulatory region, anything that binds in here we call a transcription factor. So I'm just going to call it TF, a transcription factor. And these things affect transcription. So RNA polymerase binds in here at the TATA, but there are other things that can stick to the DNA and then attach to the RNA polymerase that will either speed it up or slow it down. And this one here that attaches here, there's a special one. It's not considered an enhancer, it's considered a bending protein, and I'm going to add that one in just so we know about it, and it's called a Gaga. It attaches to the letters GAGA, like Lady Gaga. So the Gaga factor, all right, this protein right here will attach to these letters and allow this to bend over. So in this particular case, you have to have this and this attached before you can alter the speed of the transcription. So multiple transcription factors, this one and this one, bind here, whatever this one is called, and then the Gaga factor bind in between. So the Gaga would have to bind here so that this can bend over and attach down here. So this has to bend over to alter the speed of transcription. And so this is called regulation, and it isn't turning on and off like the CpG island. It's controlling the volume or the rate of transcription. And there's a question now as to whether or not we should include this in the idea of a gene, because it is absolutely inherited. And if you remember, our definition of a gene was a unit of inheritance, and absolutely this can be inherited. You change the letters here, you change, or even in the C's and G's, you change any of these letters and they're in many ways as important as the gene. So realize that this is also part of the inheritance, something that is inherited, so therefore we should consider this part of the gene perhaps. Now there is an interesting thing that you ought to know, is that we now know that if you alter the gene, all right, this part, almost always you make the protein wrong, then the cells die. So if for instance there's a mutation while you're in the womb, for instance, the baby almost always dies if there's a mutation in a gene. However, out here, not so often. You get a mutation out here, okay, you change the rate. Maybe instead of making 10 RNAs a minute, maybe you now make 100. Might be weird, but you live. You still make it. Or maybe you make too little. And what we're finding out now, and what's really interesting that I want to talk a lot about later on, is that we find that most human diseases are caused by mutations out here in this region before the promoter. All of this region out here, or actually everything in here, that's where most of your mutations are at that cause human diseases. You change the actual protein, the part down this way, and usually the cell doesn't live. There are a few diseases that are caused by that. But for the vast majority of human diseases, now that we've sequenced the genome and we've started studying this, we realize that this is the region that leads to human disease. And we're just now actually in the last 10 years started to realize this, and that's now the new idea, and that is spreading slowly through medicine and science. And the idea is we need to change our definition of a gene to include this region. And it's not in the textbooks quite, maybe in advanced graduate textbooks, but as an undergraduate, they tend to still think of it as this, or perhaps if it's a brand new textbook, they'll think of it as this. But in this class, we ought to at least consider the idea that a gene is all of this. And here's just another representation of this thing like the GAGA, which when it binds, it allows the enhancer to move over and touch the promoter. So we have to understand there are transcription factors, those are proteins, and they stick to things like these enhancers and this GAGA and all of these things. So there are all these transcription factors. Now that's different than the thing that sticks to the CpG island and methylates it. Methylation is a completely different system, a different set of proteins. We would not call this a transcription factor. This is just something that causes you to turn off the gene, or if you can take the methylation off, turn it on. So these proteins are a little different than transcription factors. They just turn things on and off. And these are usually hormones, by the way, that do this. All right, so this is our idea. I would like you to understand these parts, and we'll talk about each of these parts specifically in a video for like CpG islands. We'll talk about just CpG islands, and we'll talk about just enhancers and things like that for a different one. But I thought I would do this for you so you could get a feel for how biology is changing and how much sequencing the human genome changed our understanding of actually down to the level of, well, what is a gene?